System book that girl give to you.
Hello, students. Hello, doctors.
Yes.
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 Will you send me your free time in the morning next week? So I will arrange a meeting with you face to face in our department to take a lecture in the department. Will you send me your timetable or your schedule? What time are you free next week from next Saturday, Sunday, Monday during the next week? Which time you are free during the day?
I will send you via WhatsApp. I will send you via WhatsApp.
Okay, send me your timetable. Okay, we are going to start now. Of course, you will receive a handout. You will receive a handout from the instructors. It is supposed. I will send you also the handout. The handout is an abbreviation of your book. So you are only asked about this handout in the exam. So any additional details in the book is not required. You are going to be asked about this handout details only. Okay? Sometimes the handout refer to your book. So you also must have the book, the biochemistry department book, the original book of the department, in order to study some details in which the handout refer to. Okay? Okay,
Yes.
today we are going to finish carbohydrate chemistry, then I will shift to metabolism directly, because metabolism is much more difficult than chemistry, and chemistry, you can hear a recorded video and it is easy to understand it, but in metabolism, it needs more explanation. It needs discussion and explanation, okay? We are going to start now, polysaccharide, heteropolysaccharide. Stop sharing, mute, allow participants, don't allow participants.
Hello, doctor.
Okay. Okay. I will start now, heteropolysaccharide. In the last lecture, I discussed homopolysaccharides. Of course, you have to know the difference between amylose and amylopectin, and I have discussed this in the previous lecture. Any question you need to ask about it, I will stop share and unmute your microphone to allow you to ask about it. And also the table which makes a difference between starch and glycogen. Now we are going to study heteropolysaccharide. Hetero means different sugar units. In homopolysaccharide, we have the same sugar units attached together. We have several glucose molecules attached together, okay, to make starch or glycogen. But in hetero, there are different monosaccharides or monosaccharide derivatives attached together. For example, we may have glucose attached to a derivative, such as uronic acid or gluconic acid. Glucose attached to galactose, or glucose attached to galactose sulfate or amino sugar. These heteropolysaccharides are called glycose aminoglycans. They are termed glycose. Glyco means carbohydrates or polysaccharide. Amino means amino sugar. Of course, I have discussed amino sugar in the previous lecture, which contains amino group at carbon number 2. Glycans means also polysaccharide. They are termed GAGs. The old name is mucopolysaccharide. This is an old name. but you must know the two names, mucopolysaccharides and glycosaminoglycans or GAG. Glycosaminoglycans are unbranched long chain of polysaccharides. The long chain is usually greater than 50 sugar unit or 50 monosaccharide, usually several hundreds, 300, 400, unbranched long chain. The most characteristic feature of the GAGs is the repeated disaccharide units. What is the meaning of repeated disaccharide? We have a unit which is formed of amino sugar and uronic acid. Amino sugar and uronic acid. This is called the disaccharide unit. Of course, amino sugar is monosaccharide, uronic acid is monosaccharide. Together they are disaccharide. This unit is repeated several hundred times. Amino sugar, uronic acid, then attach it to another amino sugar, another uronic acid, then attach it to a third amino sugar, third uronic acid. This is a characteristic feature. There is repeating disaccharide units, and these disaccharide units are the same. The same amino sugar, the same uronic acid are repeated several hundred times. Glycosaminoglycans are classified either sulfate-free, sulfate means here does not contain sulfuric acid, sulfate-free. The example of sulfate-free is hyaluronic acid. The second class or category, sulfate-containing. GAGs which contain sulfuric acid, such as chondroitin sulfate, keratan sulfate, dermatan sulfate, heparin, heparan sulfate. Of course, you have to memorize the name, but there are several details. I will tell you that you are not asked to know them, okay? You have to know the name, sulfate-free. The example is hyaluronic acid. Sulfate-containing, there are several examples, about 5 examples: chondroitin sulfate, keratan sulfate, dermatan sulfate, heparin and heparan sulfate. Okay? The constituents, you are not asked about the constituent on the exam, so I will make a cross about it. The constituent you are not asked, but you will be asked about the function. You have to know the name, examples of heteropolysaccharide, and you have to know the function of heteropolysaccharides. Okay, GAGs are usually attached to a protein core, so they are termed proteoglycan. Proteo means protein. Usually, the beginning of the word proteoglycan begins by protein, means that the protein constitute less than 5%, little amount of proteoglycan. Proteoglycan are large molecule formed of protein attached to GAGs. GAGs are heteropolysaccharides. But the most prominent feature is that glycan, which is the heteropolysaccharide. Glycan constitute about 95% of the molecule of proteoglycan. But the protein minimal amount or minute amount, less than 5%. Okay? GAGs are the major component of our extracellular matrix in addition to another proteins. We are going to study them in the extracellular matrix proteins. Okay? You must know the details of the composition of proteoglycan because we are going to study another molecules called the glycoproteins. Again, proteoglycan, small amount of protein, less than 5%. Glycan means heteropolysaccharide, more than 95%. And they are major component of extracellular matrix. The most important thing is to know the functions of GAGs and the GAGs which are attached to the proteins, which are termed proteoglycans. Functions is very important in chemistry. This is the most important thing, why we study these molecules in biochemistry because they have important biological function and they are important in medicine. Okay, I will stop to ask you if this clear or not. Unmute all. Okay, I want to ask, do you hear me and everything is clear?
Yeah, we are hearing you well. But,
okay, is it clear? I advise you to hear the recorded lecture. And you also have another recorded lecture in the Blackboard. You get the account, username, and password to the Blackboard of the university. Do you know the Blackboard?
no.
You don't get, you must ask the Vice Dean about the username and the password of the Blackboard of the university. This is an online framework that has many lectures recorded and uploaded in this Blackboard in all subjects, not only biochemistry, in all study subjects such as anatomy, histology, physiology. So you must have account on the Blackboard of Cairo University. Okay, I will again mute all of you to complete the lecture. Functions of GAG. As we said, they have very important function in the extracellular matrix. Extracellular matrix contains the connective tissue. GAGs are highly charged. Why they are charged? Negatively charged. Because they contain sulfuric acid. They contain phosphoric acid. They contain uronic acid, glucuronic acid, galacturonic acid. And of course, we have studied sugar acids in the previous lecture. We have studied aldonic acid, aldaric acid, uronic acid. You have to memorize them. So any acid has negative charge, any acid. And we have sulfate here, sulfuric, we have phosphoric. Negative charge come from the carboxylic group, which is a chemical functional group that specifies all organic acids. Okay, what is the value of this negative charge? The value of this negative charge is that the negative charge attracts the hydrogen of the water molecule, trap the water molecule. What is the value of trapping the water molecule? The hydrogen is positively charged. So opposite charges attracts to each other. And water has hydrogen ion positive. The positive hydrogen ion of water is attracted to the negative charge present on GAGs or heteropolysaccharide. So water with GAGs form a gel which is hydrated. If you touch your skin, you find that the skin of your hand, your palm of the hand is soft because 75% of your body contains water. What traps water in your body? Keep water, the GAGs. GAGs attract and trap water in the connective tissue to make the connective tissue like the gel and smooth because it has numerous functions we are going to discuss. If you touch the table, which is unliving organism, you will find that the table is hard, okay? If you touch your nails is hard. So it does not have enough amount of GAGs to trap water. So this is a hard constituent of the body. But the soft constituent must trap water. And we are going to study the function of this gel. This is a jelly, gel-like substance formed of water attracted to GAGs or heteropolysaccharide. The gel provides mechanical support for extracellular matrix. The gel acts as a filter that allows diffusion of ions, such as calcium ions, and prevent diffusion of bacteria, for example, foreign cells. Prevent the diffusion of large protein molecule, but it allows diffusion of ions such as sodium ion, potassium ion, calcium ions. That's why in our body there are gels in the extracellular matrix. Inside the synovial fluid is the fluid which is found inside our joints. Because there is friction between bones in our joint. And all the time we are moving, there are frictions between different joints. Unless there is jelly-like material or gel-like material, our bones will be eroded. Our bones will be lost due to the friction. So the gags act as a lubricant. It makes lubrication and preventive frictions between bones inside our joints. Gags also act as a shock absorbance. If you study the anatomy of the eye globe, of your eye is a sphere. In the anatomy, we will find that inside your eye, there is a jelly-like material, which is called vitreous body. This vitreous body will make shock absorbent against any trauma directed to the eye. If you touch your eye, you will find it is elastic, it is flexible. It makes the eyeball resilient, okay? And all our body contains gags that help us to absorb trauma. If you jump from a high place, your bones absorb trauma. If you look at any animal like cat can jump from the second floor, from the third floor without any injury due to this gel-like material which are formed between gags and water, which attract water. The proteoglycans interact with proteins in the extracellular matrix. 90% of proteins in the extracellular matrix is collagen. We are going to study collagen in detail, in huge details in the chapter of extracellular matrix. Elastin is another protein, and there are other proteins in the extracellular matrix. GAG is attached to this matrix protein. Extracellular matrix constitute the connective tissue. And this determines the structural organization of the matrix. If you look at our bones, you will find that our bones is formed of matrix proteins, GAGs, and calcium phosphate, okay? The mineral only does not form structure or a skeleton. The structure or the skeleton which traps bone, minerals of our bone are formed by GAGs attached to proteins such as collagen and elastin. Hyaluronic acid is a type of proteoglycan. If you revise, hyaluronic acid is sulfate-free, does not contain sulfuric acid, sulfur-free. It helps development of the embryo, movement of the organs. If you study embryology and anatomy, you will find that there are migrations of the organs and the cell and the tissue during embryonic development. Hyaluronic acid helps this movement and cell migration and morphogenesis. Genesis means synthesis of the morphology of the organs, okay? Hyaluronic acid is also involved in wound repair. How it is involved in wound repair, we are going to study this in physiology. How hyaluronic acid is involved in embryonic tissue development, we are going to study it in embryology in anatomy. There are certain enzymes which are called hyaluronidase enzyme. Hyaluronidase enzyme is not found in our body because this is a destructive enzyme. Ase means hydrolysis or destruction. This enzyme destroys hyaluronic acid. Of course, I have said that hyaluronic acid is one member of the GAGs. It forms jelly-like material under your skin. Under your skin, there is connective tissue. And this jelly-like material prevents spread of bacteria. Bacteria are around us everywhere. If you look at the table or anything, you touch anything beside you, there are huge number of colonies of bacteria. Yeah, so that our universe is not sterile. What keeps our body from these bacteria? The skin. The skin prevents penetration of bacteria. If you are injured, the wound will be infected with bacteria. And under the skin, there is connective tissue or extracellular matrix, which contain hyaluronic acid, prevents spread of bacteria. but there are dangerous bacteria, certain bacteria which can destroy the connective tissue, the extracellular matrix, and can destroy hyaluronic acid because of the presence of hyaluronidase, so that bacteria can spread to the subcutaneous tissue. Another useful function of this enzyme is that the head of the sperm can penetrate the ovum during fertilization by destroying the destruction of hyaluronic acid which surrounds the ovum to help the process of fertilization. Heparin. Heparin is a very famous anticoagulant. Heparin is present also in our body. It prevents thrombin function. It is antithrombin. It actually leads to activation of antithrombin, so it prevents coagulation. There is a balance in our body between coagulation and anticoagulation, okay? We are going to go outside the room within 10 minutes and to re-enter the room. Also heparin inhibit factor number 9. Factor number 9, we are going to study coagulation factor in physiology. There are 12 or 13 factors. These are blood proteins which help the process of blood coagulation or formation of blood clot. Heparin is a natural anticoagulant. It inhibits thrombin, which is one of the coagulation factor, inhibits coagulation factor number 9, number 11. Another function of heparin is that it helps a lipase enzyme. What is a lipase? Lipase means lipid. Ase means hydrolysis or destruction, breakdown of the lipid. Lipoprotein, this is an enzyme which are attached to a lipid and protein called lipoprotein. And this enzyme makes hydrolysis of the lipids which enter inside the blood after absorption, after digestion, absorption, lipids will be absorbed and go to the systemic circulation. This enzyme helps removal and clearance of the blood lipids. I will stop share to ask you if you understand or not. Okay, is everything clear? Do you hear me well?
Yeah, we hear you well.
Okay, anyone who has any question, please write it in the chat box. Okay, if you have any question.
Okay, before we get out, can you repeat please a little bit of the function? The last one, number 5.
Of which one? Function of which
Ephrin.
one?
Ephrin. Function of gas.
Can you write its name because I don't hear well? Can you write the name? What type of gags
Okay.
you need the function? Write it in the chat box, please.
Mesh. I'll write it. Function of
Thinners or thrombin? This is not thinner. This is thrombin. Okay. I will ask you now. I will ask. We are going outside this room within 7 minutes. Within 7 minutes, we are going to go outside the Zoom meeting and then we re-enter again by the same credentials, okay? Because the Zoom meeting will stop after 7 minutes and then we are going to enter again. Every one of the gags has function. You have to study and memorize it. Heparin is a natural anticoagulant present inside our blood because there is balance between coagulation and anticoagulation. If there is no balance and coagulation factor win the battle, so all your blood will be coagulated and this will be fatal. Blood clot is fatal. If it occurs in the heart, in the brain, this is fatal. If you don't have coagulant and there is anticoagulant, you will have bleeding tendency. You will have internal bleeding or external bleeding. So there is a balance between coagulation factors and anticoagulation factors. So there is a balance, okay? Coagulation factors are 13 factors. You are going to study them by name and by number in physiology in great detail. This is not the topic of biochemistry. One of the anticoagulants is heparin. Heparin inhibits factor number 2, which is thrombin. Heparin is anti-thrombin. Anti means against. Stop the function. Stop the function of the thrombin. Thrombin help blood coagulation. Heparin inhibit the function of factor number 9. Inhibit the function of factor number 11. These as regard coagulation. Heparin had another function. Heparin activate lipase enzyme, but it has a long name. Lipoprotein. Lipo means lipid. Proteins means lipid attached to a protein. These are the particles of lipids which are found inside our blood. You are going to study it in lipid metabolism. Protein enter inside the systemic circulation, lipids enter the systemic circulation surrounded by a protein in the form of spherical particle. These lipids which enter our blood through the process of digestion and absorption from the intestine, the lipid passes to the liver, then passes to the systemic circulation. It must be cleared, removed from the blood. Lipoprotein lipase break down the lipids into fatty acids, so adipose tissue can uptake these fatty acids. Other organs which need the fatty acids, such as cardiac muscle, utilizes fatty acid for energy, can uptake it. So lipoprotein lipase breaks the lipids, which are triglycerides, to liberate fatty acids. Fatty acids are taken by the tissue to get energy, oxidation of fatty acid to get energy. Heparin activate these lipase enzyme, which are termed lipoprotein lipase enzyme. Keratan, you have to know the name. Keratan, it is very approximate the name of the cornea. Do you know the cornea? The cornea is the transparent body present in the front of your eye. If you look at the eye of your colleague, you will find that there is a circle which is colored either black or brown or green or blue. In the European population, you will find that this circle is colored blue. This is called iris. The colored circle is called iris of the eye. There is a transparent body found inside this colored circle, inside this colored iris, which is called a cornea. The cornea must be transparent. The transparency of the cornea is very important for vision, because if the cornea loses transparency, human will lose its sight and he will be blind. So keratan sulfate is important for keeping the transparency of the cornea. How this occurs, this is not your topic in the undergraduate. How protein will be transparent, how GAGs, polysaccharide, will be transparent. This is not important for you in the undergraduate study. We have studied heparin. There is another structure which is similar to heparin but termed heparan. Heparin has I. Heparin has I letter. But heparan has A. Heparan sulfate. Of course, it is proteoglycan. Glycan means polysaccharide, 95% carbohydrate, and 5% protein attached to polysaccharide. Heparan is found in the plasma membrane. The plasma membrane has very important vital functions because receptors of various kinds, hormone receptors, channels such as calcium channels, sodium channel, potassium channels, neurotransmitters receptors which accept neurotransmitters. So the structure of the receptor contains proteoglycans. One of these proteoglycan is heparan sulfate. This receptor helps cell communication. Cells communicate with each other. Cardiac muscle cells communicate with each other, so they all contract at the same moment and they all relax at the same moment, synchronous actions. So cell-cell communication or interaction occurs through the cell membrane receptors which contain proteoglycans. Aggrecan found in the structure of the cartilage. This is a very complex structure, enter in the composition of the cartilage. You are going to study cartilage in anatomy and histology, and the function of the cartilage is very important to protect the bone from friction. The last thing we are going to study in this chapter of carbohydrate chemistry is glycoproteins. As you see the name, glyco means carbohydrate. the first part of this molecule is very small, is less than 5%. يبقى carbohydrate here is little. Protein is more than 50%. Protein is the major composition of glycoprotein. This is the opposite of proteoglycan. Proteoglycan, the major composition is carbohydrate. 95% of proteoglycan is carbohydrate. Here, protein is the major composition. Can reach from 50% to 90%. Proteins attached to oligosaccharide chain. What is the difference between glycoproteins and proteoglycan? If you look at the structure of GAGs or proteoglycans, as I said before, GAGs has disaccharide units. Amino sugar, uronic acid attached together. For example, glucosamine, amino sugar attached to glucuronic acid.